Good afternoon, everyone. Welcome to the Allen School Colloquium. It gives me great pleasure to introduce Matt Golub. Matt is a postdoctoral fellow at Stanford University, working at the intersection of machine learning, neural engineering, and systems neuroscience. And he received his PhD in electrical and computer engineering from Carnegie Mellon, where he developed brain-computer interfaces. And these were interfaces for scientific inquiry, so really special kind of interfaces. And his research has been published in many prominent journals. So for example, Nature, Proceedings of the National Academy of Sciences, Nature Neuroscience, as well as in machine learning conferences like NeurIPS and ICML. And his work has been recently recognized by the prestigious K99 Pathway to Independence Award from the National Institutes of Health. And today, Matt will be telling us about his research on reverse engineering computation in the brain. And just as in terms of the questions that you may have during his talk, please type the questions in chat and I'll let Matt know. Or you can also wait for Matt to stop for questions at appropriate junctures in his talk or at the end of his talk when the floor will be open for broader questions. So take it away, Matt.
Great, thank you so much for that wonderful introduction, and it's a real pleasure to be with you all today. So I'm a computational neuroscientist. I'm broadly interested in the intersection between machine learning and neuroscience. And I'd like to just start off by pointing out a few of the amazing things that our brains do for us. So this rock climber is deciding where to move her hand next. She sees a great hold out to the left, but she also sees a nice foothold out to the right. So her brain is just awash in computations, sensory perception, evidence accumulation, movement preparation. And these computations serve higher level brain functions that she's engaging. Functions like feedback motor control, decision making, and learning. And in systems neuroscience, we want to understand how our brains give rise to all of this and more. How do we do this? Well, we record from neurons in the brain. And if you record the electrical activity of a single neuron in the brain, you see a voltage signal with these high amplitude events called spikes. And these spikes represent a fundamental unit of information in the brain. So when we study neural activity, we often reduce these noisy voltage signals just down to the time of each spike. And with modern technology, we can record simultaneously from many neurons. So here, each tick mark represents a spike from a single neuron at a given time. So I'm a computational neuroscientist and I develop machine learning tools to reverse engineer computations in the brain. I'm interested in questions like, how can we study neural activity to infer the computational principles underlying our brain's functions? What are the algorithms implemented by specific populations of neurons in the brain? And how should we design our experiments and analyses with hypotheses about computation in mind? So here's a quick overview of my work to date. In graduate school, I developed brain-computer interfaces that translated neural activity from a monkey's motor cortex into movements of a computer cursor, all in real time. So I did some engineering work improving the performance of these systems. And then I saw an opportunity to use these systems for basic scientific studies, for reverse engineering brain computations related to feedback motor control and learning. And my more recent work focuses on how we can reverse engineer computations through neural population dynamics, which describe how neural activity evolves over time. And these population dynamics often reflect structured dynamical motifs, attractors, saddle points, oscillations, which can compose to perform computations in the brain, computations like evidence accumulation and movement preparation, for example. And I'm currently using this computation through dynamics approach to study perceptual decision making in the brain. So I'm quite excited to share with this particular audience because neuroscience benefits tremendously from connections to computer science and engineering. So for example, this work has involved developing and applying techniques in control theory, dimensionality reduction, dynamical systems, convex optimization, probabilistic models, network models, and deep learning. And for those of you who might be new to neuroscience, I'd encourage you to think throughout the talk about other potential bridges with the techniques that you work on. And I'd love to hear from you if you're interested in getting involved. So this is the outline for my talk today. I'll briefly talk about each of these projects, and I'll take a deep dive into learning and computation through dynamics. And then I'll close with my future plans that have been enabled by this work. So let's jump in. So brain-computer interfaces translate neural activity from many simultaneously recorded neurons into control signals for driving a prosthetic device, like a computer cursor or a robotic device, or for electrical stimulation of muscles in the arm. And most recently, BCIs have been able to decode human speech and handwriting. The systems that I've developed use a 96-channel Utah array that's surgically implanted directly into the motor cortex, primary motor cortex, or M1, of a monkey. And early in grad school, I worked on algorithm design for improving the performance of these BCI systems. So here's a video. I hope it plays smoothly for you all. Here the monkey is using his neural activity to drive this green cursor out to these blue targets. And in this work, I developed variations on a Kalman filter for mapping neural activity into velocity of the cursor. I also conducted these monkey experiments, so I have some firsthand experience about the complexities of neurophysiology, experiment design, and behavior. A primary motivation now for BCIs is to help people with spinal cord injuries or neurodegenerative disease, people who have lost the ability to move or to communicate. And BCIs offer to restore some of these capacities for movement and communication. But early in grad school, I saw an opportunity, an area of untapped potential, and that was to use BCI for a different purpose, to use BCI as an experimental paradigm for basic scientific studies about the brain. And this new take on BCI allowed me to reverse engineer computations used in the brain for feedback motor control and learning. So in one line of work, I developed probabilistic inverse optimal control methods for identifying what is the monkey's internal model of this BCI mapping. And there I found that a mismatch between the animal's internal model and the actual mapping explained a majority of the monkey's mistakes. In another project, we asked, how does the brain choose which control signals to send when many different control signals redundantly map to the same cursor movements? We also asked, how do cognitive states influence behavior? And how does neural activity change during learning? Now I want to take one of our deep dives and talk a bit more about this last project on learning. So here we really took advantage of the fact that in BCI, we choose this BCI mapping, and we can change it to ask the animal to learn to produce different patterns of activity. So we designed a learning experiment where in each session, we began by using an intuitive BCI mapping that was easy for the animal to control. And then we abruptly switched to a perturbed BCI mapping that was initially challenging for the animal to control. And then we got to watch and record as the animal learned to control this perturbed mapping. And the whole time the animal is doing this center out cursor task, just like I showed in the video, but in case that didn't come across smoothly, the animal's task is the cursor starts in the center, then a green target appears at the periphery, and the animal needs to modulate his neural activity that's being recorded in order to drive the cursor out to that green target. So to start, I want to introduce a key abstraction for this portion of the talk, which is a neural state space. And so in this space, each axis indicates the firing rate of one neuron, and a point in the space indicates the firing rates across all the neurons in a short time window. And here we can only visualize 3 neurons, but really the dimension of the space is the number of simultaneously recorded neurons. In this line of work, we were motivated by the observation that we and many others have made, which is that population activity patterns don't uniformly fill the space, but rather they tend to lie on a low dimensional subspace that I'll refer to as the intrinsic manifold. And when we learn something new, we should see changes in this neural space. So we became curious about the role that this manifold structure plays in our ability to learn, in our ability to make changes to neural activity in this space. In particular, how does the structure of population activity constrain our ability to learn? So we designed our experiment precisely with this question in mind and with this concept of the intrinsic manifold and dimensionality reduction in mind. So in each experimental session, on each day, we began the experiment by using factor analysis to identify this manifold. And then we defined an intuitive BCI mapping that lies within this manifold and was easy for the animal to control. Now here for visualization, I can only show you this three-dimensional neural space with a two-dimensional intrinsic manifold and a one-dimensional BCI mapping that decodes a one-dimensional cursor velocity, so just left and right. So in this visualization, activity patterns to the left of this dashed line would drive the cursor to the left at increasing speeds, and activity patterns to the right of the dashed line would drive the cursor to the right. Okay, but recall in our experiments, we're recording from a 96 channel array. So our neural space is actually 96 dimensional. Using factor analysis, we found that a 10-dimensional intrinsic manifold best described the data. So that's what we used. And as I mentioned, and as I showed in the video, we decoded two-dimensional cursor velocities. So not just left, right, but also up, down. So in one set of experiments, we then perturbed that BCI mapping. We changed it, rotating it within the 10-dimensional intrinsic manifold. So this was asking the animal to reorganize activity within the intrinsic manifold. Okay, now where a different set of activity patterns drive the cursor to the left and a different set of activity patterns drive the cursor to the right. Okay, so the key here is that the new mapping is still within this intrinsic manifold. Then in a second set of experiments, we changed the BCI mapping so that the new mapping lies outside of the manifold. And this is asking the animal to learn to generate activity patterns outside of the manifold. And what we found was the within manifold perturbations were much more learnable on the 1 to 2 hour timescale of our experiments, meaning learning is faster within the manifold. Here's a representative within manifold session where I'm showing acquisition time, which is how long it takes for the animal to move the cursor from the center to the target. And under the intuitive mapping, this is stable and relatively fast, around 1 second. Control is quite good. These are some example cursor trajectories. Then when we switch the mapping, we see an initial hit to performance. But then with learning, the animal's behavior improves, although he never gets quite back to where he was under the intuitive mapping. And I'll come back to this point later. By contrast, here's a representative outside manifold session. After the mapping is switched, performing never really improves. We don't really see much learning. And this was the general trend that we saw across many experimental sessions. The amount of learning was greater within manifold compared to outside of manifold. So this really begged a number of new questions. First, could animals learn outside of the manifold if given more time? And we found there that indeed they could, given roughly 10 days of practice with a given outside manifold BCI mapping. And second, how do neural activity patterns change during the learning that we did see? So I'll spend the next few minutes diving into the neural activity to answer this question. And here we'll focus on the within manifold perturbations because those induced the most learning. So let's dive into the data. So recall we began each session with an intuitive BCI mapping, which was easy for the animal to use right off the bat. So that means when the animal intends to move to the left, he generates activity patterns that for the most part drive the cursor to the left. And when he intends to move to the right, he generates a different set of activity patterns which tend to move the cursor to the which do move the cursor to the right. But after we switch the perturbed the mapping and before any learning has happened, Now when he intends to move to the right, some of these patterns still move the cursor to the right. That's good. But some of these patterns now move the cursor to the left. That's bad. And vice versa. So how did the animals learn to fix this? That's the question we wanted to answer by looking at the neural activity. So we started with a few hypotheses. Our first hypothesis we call realignment, where the animal realigns these clouds of activity with the new mapping, realigning them to maximize behavioral performance through this new mapping. And in a moment, I'll show how this hypothesis is not consistent with the data, which instead support a second hypothesis, which we call reassociation. And here the animal simply repurposes existing activity patterns. So in this visualization, this just corresponds to recoloring all of the patterns. So here, the animal doesn't change the overall repertoire of the activity patterns, but simply instead reassociates each activity pattern with the movement that it actually causes through the new BCI map. So again, this is the before learning data. This is what we predict with realignment. This is what we predict with reassociation. And we found this concept of the neural repertoire to be really critical in how we tested these hypotheses. So we defined the neural repertoire simply as the region in neural space where the before learning activity patterns reside. If I just copy that same repertoire on top of realignment, you see that realignment allows for changes to that repertoire. There are some points outside of this gray shading, whereas reassociation does not allow for that, but rather it does the best that it can with a fixed repertoire. And the best that you can do with a fixed repertoire is just recolor the points. Only choose these points up here when you want to move to the left. So to begin testing these hypotheses, we looked for this concept of repertoire change in the data. To do this, we devised a repertoire change metric based on nearest neighbor distances to activity patterns in the before learning repertoire, in the before learning data. And we normalized this so that a value of 0 indicates no repertoire change, which you would find for this blue activity pattern, which looks like it's just in this repertoire. And a positive value would be found for out of repertoire activity patterns like this red pattern. And when we did this computation on the neural data, looking at the neural activity patterns that the animal produced after learning these mappings and comparing that data to the before learning repertoire, we found that the data indicate no change in the neural repertoire after learning. Okay, so we'll come back to this in a second, but first I want to go a bit deeper. Now, so far we've been looking at a lot of pretty pictures based on qualitative hypotheses, but a more complete understanding of the data requires that we make these hypotheses quantitative. So to do this, we built models to predict the distribution of after learning activity patterns that we should expect under each hypothesis. So these predicted distributions were the solutions to hypothesis-specific convex optimization problems. For realignment, we found the activity patterns that would maximize behavioral performance through the perturbed BCI mapping, okay, subject to physiological limits on each neuron's firing rate, realistic levels of neural variability, and all activity patterns needed to reside in the intrinsic manifold. Okay, for reassociation, the optimization was identical with the exception of one added constraint, that all the activity patterns must be within the before learning neural repertoire. So these models allowed us to simulate entire neural data sets consistent with each of these hypotheses, which then allowed us to compare against the data across a multitude of different measures. So first, returning to our repertoire change metric, realignment predicts indeed this repertoire change, whereas reassociation matches the data, both in terms of having an average repertoire change of 0 and in terms of variability across data sets. Reassociation also matches the data in terms of the extent of behavioral improvements we observed after learning. And so this offers a concrete explanation to why performance never returns to intuitive level performance. And by contrast, realignment predicts even more behavioral improvement than the animals showed. We also looked at a multitude of different measures of neural covariability. And finally, I've only had time to share two of our hypotheses, this realignment and reassociation. But we also looked really carefully and quantitatively at a collection of hypotheses, which ultimately were not consistent with the data. So to summarize, we first asked, how does the structure of neural activity constrain learning? And there we found that it's relatively easy to change activity within the manifold compared to being more difficult to generate activity outside of the manifold. This manifold reflects population level correlations, which likely reflect connectivity within the brain's network. So taken together, these results suggest that it might be difficult to change network connectivity on these 1 to 2 hour timescales. I mentioned that outside manifold learning is possible on longer timescales of about 10 days. Then we wanted to understand, how does neural activity change during these single day within manifold experiments? And there we discovered that learning proceeded by reassociating existing activity patterns with different movement intentions after learning. And we thought this was a bit surprising because there is a better solution, which is our realignment hypothesis. And the animals didn't use that. One exciting add-on that I'll mention is that we recently found that many of these principles of learning that we discovered in the BCI context also play in motor learning in outside of the BCI context in our movements paradigm. Okay, so this may be a good time to take stock, take any questions before we move on to the next section.
So this is more of a clarification question, but since I work in human-computer interaction, you worry a lot about the design of experiments and your subjects. What leads the monkeys to be motivated to perform well on this task? Because obviously that's their motivation, such as it is going to be a key part of interpreting what you're seeing here.
Yeah, that's a great question. And you're identifying one of the most important aspects of monkey research, which is keeping the monkey happy. Monkeys can be really, really, really hard workers, and often in many cases harder workers than humans when we put ourselves in the rig to do these experiments. So they're really motivated to get rewards. We reward them by giving them juice or other treats each time they get a successful trial. So they really want to hit the target, and they want to hit the target as many times as they can each minute. Yeah, you will hear stories about researchers who come up with really, really difficult tasks that they want to teach monkeys how to perform. And these issues of motivation are like really at the heart of, can you do this experiment? Can you get the animal to be interested in doing that?
There are a couple of questions in the chat. I don't know if we can answer all of them, but the first one there is what biologically plausible plasticity rule would allow the neural reassociations that you saw? This is from Alexandra Paye.
Yeah, that's a great question. We can't really say too much about that from just the data that we have, but these results about reassociation kind of suggest that the fastest learning processes that our brains use involve kind of just higher level changes of commands, where in this example, that would be probably like changing the inputs to the motor cortex as opposed to reconfiguring the circuit in motor cortex. But if you kick that upstream so many times, eventually, I think there needs to be some place that's making kind of rapid timescale changes. And so there may need to be some mechanisms in the brain for having really reconfigurable systems that maybe don't require changes in synaptic connections in order to have functional changes in what the brain does.
Cool. I think there's a clarification question in terms of the neural activity being measured via what particular mechanism. So can you just speak briefly about that? How was the neural activity measured?
Sure, yeah, we recorded with a 100 electrode array that was surgically implanted into the motor cortex. And so there we read out spikes, individual spikes from individual neurons, about 100 of them simultaneously recorded.
Great. And then another question from Dustin Samarli is what are the possible reasons why realignment is not used by the animals despite better predicted behavioral outcomes?
Yeah, so I suspect that this is because realignment requires changes in the connectivity of the circuit. And there's some really exciting work that maybe my lab will pursue and others are thinking about as well, which is, can you build a network model of this phenomenon? And is it possible to see realignment without changing connectivity within the circuit?
Okay, we got a bunch of questions, but I think maybe we'll hold off until the end. So I would appreciate if people come back to these questions again, so Matt can move on to the next part of his talk. Is that okay, Matt?
Yeah, wonderful.
Okay.
Yeah, thank you for these questions. Yeah. Okay, so now I want to switch gears and describe another reverse engineering approach that I've developed, which aims to understand computations in the brain through the network dynamics of neural circuits. So what do I mean by dynamics? Well, so far we've been talking about neural activity patterns as a point in a neural space. But these population activity patterns, they don't exist in isolation, but rather they evolve lawfully over time as a neural trajectory. So neural population dynamics govern how this neural trajectory unfolds over time. These dynamics are often very structured, such that if you look at the neural trajectories from many different repetitions of an animal performing some behavior, so many different single trials, the neural trajectories all reflect a shared dynamical motif. In this case, the neural circuit gives rise to an attractor motif. All of the trajectories are attracted to a stable fixed point in the neural population dynamics. Other examples of dynamical motifs include a line attractor or a saddle point or an oscillation. These are simple dynamics which can be described by a linear system. And these simple motifs are interesting because they're the building blocks of richer dynamical computations. So what do I mean by dynamical computations? Well, dynamical computations are computations that unfold over time. A simple example is a 1-bit memory, and that can be implemented by a composition of 2 attractors. So here these 2 points represent the 2 possible states of the memory. And we can visualize this dynamic either as these flow fields or as an energy landscape, where following a particular flow field corresponds to descending the energy landscape. So we now need a nonlinear function to describe this system, where now I'm also including an input u, which could be used to switch from one state to the other. So here we have a computation maintaining a memory over time, implemented by a composition of dynamical motifs, these 2 attractors. So just briefly taking stock, I have shown how neural activity evolves lawfully according to neural population dynamics. These dynamics reflect structured dynamical motifs, and these motifs can compose to perform computation. Okay, I showed this one simple example, maintaining a memory. And in a few minutes, I'll show a few more interesting examples, accumulating evidence and preparing a movement. But first, I want to share the high-level vision here. And this is where I saw another big opportunity, which is to develop an approach to identifying computations in the brain via these dynamical motifs directly from neural data. So since the 50s and 60s, researchers have been building network models with brain-like features. They can store memory, they can compute. And it's long been hypothesized that these types of dynamics support computation in the brain. But no one has been able to definitively show these properties in real neural data. That's what I aim to do. And this question represents a major theme that my lab will pursue, both in terms of developing machine learning tools, as well as collaborations with experimental groups. And this is hard. Tackling this problem requires rich nonlinear dynamical models of our noisy neural data. These models have only really made their way into neuroscience in the past few years. These models can be non-identifiable, meaning many different models often explain the data equally well. And this is an issue because we want to be able to interpret these models. We want to be able to parse out the dynamical motifs and understand how they subserve a computation. So now I'm going to walk through how I've approached these challenges. So I've been approaching this problem by thinking about a neural population as a dynamical system whose activity unfolds over time due to how these neurons are connected and due to inputs to those neurons, which might come from nearby neurons or other brain areas. Deep learning now enables us to build rather sophisticated models of these dynamics from single trial population recordings. I've spent my postdoc developing and interpreting a set of these models, which are based on recurrent neural networks, or RNNs. And we can now train these RNNs to recapitulate single trial neural recordings by learning a nonlinear dynamical system, along with the single trial inputs needed to drive that system to reproduce the data. So the core concept here is that if we want to identify the computation performed by a neural population, and we think the population dynamics implement that computation in the brain, then if we can build a rich model of those dynamics, we should be able to reveal that computation embedded in these dynamical models. Specifically, the models I've been developing are based on LFAS, or latent factor analysis via dynamical systems. And the goal of LFAS is to reconstruct single trial neural recordings by way of single trial dynamics. So here, each page represents a single trial of neurons by time. And of course, we have many different single trials. And LFAS will try to explain these spike counts under a Poisson noise model via a set of inferred non-negative rates, one time series per neuron per trial. And these rates reflect a readout from an RNN after it's passed through a few additional layers that simply function to convert continuous valued activations in the RNN to non-negative rates because firing rates cannot be negative. And LFAS will infer an initial condition for each trial and can be driven by time-varying inputs for each trial, which can either be inferred from the neural data in an unsupervised manner, or we can use other observed data as inputs. So why should we care about LFAS? Well, first, it represents the state of the art in machine learning tools for modeling single trial neural population data. And second, if we're confident in the model, then we can leverage the model as a surrogate for the neural circuit itself, allowing analyses that we simply cannot perform on a neural population. So from here on, I'll just use this simplified graphic to depict LFAS, and we'll know that it means kind of all of that complexity that I just unpacked. So far, I've been developing these techniques around a study of perceptual decision-making. Perceptual decision-making is really interesting for studying computation because it requires several connected computations. So consider this classical moving dots task from Bill Newsom's lab. In this task, a monkey watches a collection of noisy moving dots on a screen and then needs to decide whether those dots are in aggregate moving more to the left or more to the right, and then report that decision with an arm reach to the left or to the right. And this requires several connected computations, including motion perception, evidence accumulation across time, and movement preparation. So now suppose we record from some part of the brain that's modulated by this task. This core goal of mine is how can we identify the computation performed by that population just based on these single trial neural recordings. How are we going to do this? Well, we're going to lean on a fixed point analysis technique that's well established for a simpler class of RNNs, which are trained to solve tasks as opposed to trained to reproduce neural data like we've been talking about. So this fixed point analysis technique decomposes the nonlinear dynamics of the RNN into a collection of our interpretable dynamical motifs, right? Each describing a local linearization of the RNN dynamics at an important operating point of the state space of the RNN's hidden units. So this effectively reverse engineers the RNN, revealing the computation that it performs by way of a composition of dynamical motifs. So let me give you an example. We can train a task RNN to perform evidence accumulation. To do this, we would take a time series stimuli, which could represent those moving dots, with positive values indicating rightward motion of the dots and negative values indicating leftward motion of the dots. Then we would train a task RNN to spit out a +1 or a -1 at the end of each stimulus, depending on whether that stimulus was more positive or more negative. And if you do a fixed point analysis of this RNN, that will reveal a line attractor, which is the dynamical motif that performs evidence accumulation or integration. So a line attractor has stable attractor dynamics along all dimensions except for one, which is the dimension of this line. And along the line, the dynamics are marginally stable, meaning the network activity stays constant unless it's kicked up or down the line by an input. So at any given moment, the state along the line represents the cumulative effect of all previous inputs. And since the input is an evidence signal, the line attractor implements evidence accumulation. Let me give another example. So related to our decision-making task, the task doesn't just end with +1 or -1, but the animal needs to report his decision with an arm movement. So we can train a task RNN to perform this movement report. To do this, we would feed the decision output as an input to this new RNN, along with a go cue indicating when the arm should move. And we would train this RNN to produce multidimensional muscle activations, or EMGs, that would drive a choice-dependent arm movement, a reach to the left or a reach to the right. And the fixed point analysis of this RNN will reveal a single attractor whose position in state space moves with the input, with this decision input. So this is before the go cue is given. And after the go cue is given, that go cue reconfigures the dynamics to drive choice-dependent oscillations. So one oscillation in the left part of the state space and one oscillation in the right part of the state space. And these serve as a basis for the oscillatory components in these muscle activations. So taking stock again, this fixed point analysis technique decomposes RNN dynamics into interpretable dynamical motifs. I trained RNNs to perform 2 decision-related computations, evidence accumulation and movement report. And this showed us again how dynamical motifs can compose to perform computation. The line attractor performs the computation of evidence accumulation, and this input-driven attractor reconfigured into choice-dependent oscillations. These are the motifs that compose to perform a movement report. So now imagine that you could do this reverse engineering not on a simple task RNN, but on one of these models of single trial neural dynamics. I think this would be a complete game changer for systems neuroscience, because if you identified the motifs in these dynamics, which are constrained to reproduce the recorded neural activity from the brain, you would discover the computation performed by your population of neurons, and you would have found this directly from the neural data. So as I mentioned, this is hard. In particular, upgrading these fixed point techniques from these task models to these more sophisticated models of single trial neuropopulation dynamics. This requires handling the more sophisticated architectures required by these networks. These data are noisy and high dimensional compared to the relatively tame data that are used to train these task models. And how to properly identify the inputs. And this is really important because there's always a wide range of inputs that could all equivalently reproduce the data. So I'll come back to this in a few minutes. So I'm going to walk through a demonstration of the technique applied to synthetic neural data, where we have ground truth knowledge of the underlying dynamics and the computation implemented by the dynamics. So let me walk you through what I'm going to show you. So I'll start by training a task RNN to perform evidence accumulation. Then I'll generate a synthetic neural data set from that task RNN. Then I'll fit LFAS to the synthetic data set. And then I'll reverse engineer LFAS to reveal the computation performed by the synthetic neural population. And this is meant to be a proof of concept of, can we do these reverse engineering techniques on these models of single trial neural data in a controlled context where we have ground truth knowledge about what the underlying computation and dynamics are. So we've already talked about how to train a task RNN to perform evidence accumulation. We feed it many instances of stimuli and train it to spit out a +1 or a -1 for each stimulus. I told you that a fixed point analysis of this RNN will reveal a line attractor, but I only showed you in cartoon form. So let me now show you what that actually looks like, so we know what we should find when we go into LFAS. So here we're looking at the space defined by the top two principal components of the network activity. In green are single trial trajectories of the network activity on the +1 trials. And in purple are the same for the -1 trials. The X's are the first time step of each trajectory. So everybody starts out here and then goes one way or another. And this is the fixed point analysis. In black are attractors, which are stable fixed points in the dynamics. And in red are saddle points, and they're just slightly unstable in this case. But the most important feature here is that these fixed points all fall on this one-dimensional manifold, and they coexist in the absence of input to the network. So what this means is that in the absence of input to the network, when the input is 0, then the network state relaxes onto this curve. And the effect of the input is to kick the network state along the curve. So this is effectively a line attractor. And this is the ground truth dynamic that we hope to discover using LFAS. Now let's generate a synthetic neural data set from this RNN. To do that, we use the time varying rates in the RNN to specify inhomogeneous Poisson processes from which we can generate spike counts. So this is a synthetic data set that's formatted just like our real single trial neural population data. But here we know that the synthetic population that generated these data was performing this evidence accumulation computation. And we have the line attractor dynamics to prove it. So then we fit an LFAS model to this synthetic data set. And I had mentioned that a key challenge is how to identify the inputs. This is crucial because systems with different inputs will learn different dynamics in order to transform those inputs into the data. Okay, so here I fit several different models, each with a different type of input. One model used the actual ground truth inputs, but other models used other plausible inputs, for example, the output of the network, which actually would explain the data rather well because these hidden neurons generated these these hidden units generated these outputs. And then I used model selection guided by reconstruction error on the neural data. And that correctly identifies the correct inputs, which are these stimuli to the network. And now this is where the payoff comes. Now we reverse engineer the trained LFAS model. So on the left, I'm reminding you what we found in the task RNN. And this is the ground truth dynamic that we hope to recover. Okay. And this is what we find when reverse engineering the LFAS model. It's also an effective line attractor. And while it looks a bit messy relative to the ground truth dynamic, it, I think, rather impressively identifies these basins of increased stability separated by regions of slight instability all along the curve. So not only did LFAS recover the line attractor motif, but it even recovered some of these finer details. And we did this directly from data. Yes, it was synthetic data, but it was still noisy single trial spike count data. So I had mentioned that a key challenge here is how to identify the inputs. I mentioned a bit about the approach here, but there are other approaches. The simplest approach is what I used, and that's a hypothesis-driven approach where we specify candidate inputs, then we use the reconstruction error on the neural data as a guide. So the better you can reconstruct your data, the more confident you can be about the inputs that you used and the corresponding dynamics learned. Okay. We can also take an unsupervised approach and infer the inputs from the data. Okay. And here you would specify a prior distribution for the inputs, which is used to encourage the model to learn simple inputs rather than passing every spike in the data in as an input. And finally, in both approaches, the inputs are only identifiable down to an invertible transformation. And this is a caveat that needs to be carefully managed in any study. Okay. So to summarize the past few slides, we fit LFAS to the synthetic neural data. Okay. Then a fixed point analysis of LFAS recovered the ground truth dynamic, this line attractor, and the computation that the line attractor performs, which is evidence accumulation. I don't have time to go through it, but this also works for other computations, not just the line attractor. For example, we can use it to recover these ground truth dynamics in the case of fitting LFAS to data generated from that movement report RNN that I showed earlier. And altogether, this is a proof of concept. We can use these techniques to reveal computations from noisy spike count data in these controlled settings. And this gives us confidence now to go out and apply these techniques widely across real neural data sets. And popping up one more level, in this half of the talk, I showed how neural activity evolves lawfully over time according to neural population dynamics. These dynamics reflect structured dynamical motifs, and these dynamical motifs compose to perform computation in the brain. I showed you a few examples from computations for maintaining a memory, accumulating evidence, and preparing a movement. And we can now fit rich nonlinear dynamical systems of noisy neural data, and reverse engineering of those models is beginning to enable data-driven identification of computations from neural population dynamics. So I'll pause for another few minutes for questions before jumping into some future directions.
Yeah, I think Jacob has a question.
Super cool work. I was wondering, okay, so I had two questions and maybe we should save this for the end, but just ask it now. So one question is, I know there's been some work on learning like latent representations of neural activity, and would it make more sense to maybe, or not maybe make more sense, but maybe make the problem more tractable instead of like looking at the hidden state of an RNN, but to learn like dynamics in some like learn latent space and analyze those dynamics. And two, all of this work looks like rate encoding, but is there like, I know that there's some work in like temporal encoding, like the actual timing of the spikes itself. Like, are there ways of kind of accounting for this information? Like, is that something that is like interesting to look at?
Sure. So for the last question, the rating or the timing of spikes is not something that we've looked at, or really this type of rate-based modeling can speak to. But it's certainly important in some corners of neuroscience, for instance, in auditory coding. In terms of the latent representation, I agree with you that looking at lower dimensional spaces is often easier. And there are some aspects of these dimensionality reduction concepts embedded in these models and can certainly be dialed up if that's helpful. But often, in order to have a model that's complex enough or has a high enough capacity to describe complex data, you need to have a complex hidden space. So if you crank down the dimensionality of the hidden space, the number of hidden units in these networks, you will run into optimization problems where at some point you can no longer explain the data. So that may be a trade-off, and I think is part of the reason why this is exciting and difficult to wrangle these large networks.
Thank you.
I think in the interest of time, I'm going to skip this next section on decision making, but basically we're applying this to data from monkeys who are performing this decision making task, and preliminarily it's telling us that it looks like this area of the brain, PMD, that we're looking at is not performing an evidence accumulation computation, even though we see signals that represent accumulated evidence in that brain area. And this is kind of a result that's really only enabled by this modeling approach of looking at single trial neural dynamics. So now let me take a step back and describe how this work sets the foundation for my future plans. So the overall vision for my lab will be to develop these dynamical models as surrogate neural circuits. We can build these models with data from any organism performing any task, recording in any brain area using any recording modality. And if our model is sufficiently expressive and fit to the right data, it can become a surrogate for the actual neural circuit. The fixed point analysis is just one example of this. And this is such a timely pursuit because these deep learning models truly thrive the more data we feed them. So as new recording technologies are emerging and becoming more accessible to experimental groups, these modeling techniques are really well poised to usher in a new era of big data, deep learning neuroscience. So I'll briefly describe, I listed 3, but I think I'll skip one in the interest of time. My initial big projects that are aligned with this vision. And for each of these projects, they each involve solving core machine learning problems. This will be the focus of my research group, and I'd love to work with other machine learning groups at the University of Washington in these directions. These projects also address cutting-edge problems in neuroscience, and my group won't require animal facilities or wet lab space, but rather I have several ongoing collaborations with experimentalists, and I'm eager to form new collaborations as needed. So first, I'll be really unleashing this computation through dynamics framework broadly across many real neural population data sets. I've only recently developed these computation through dynamics techniques, and so far I've mostly just vetted them on synthetic data. And now it's prime time to take these fresh new tools to discover computations performed by many different neural populations all over the brain. So I have some existing collaborations in place to look at computations in prefrontal cortex during decision-making and problem-solving tasks. And I'd also be interested in creating new collaborations across UW, maybe with Amy Osborne to study computations for neuroplasticity, or Elizabeth Buffalo to study computations related to memory, or with Nick Steinmetz to study distributed computation across brain areas. And there are many others in this space at UW. And there's also lots of exciting machine learning work to be done here related to how to properly identify the inputs and introduce new latent variable methods that can be robust to hypotheses that we might have that are kind of partially specified, and we want to allow the model and the optimization to improve upon our guess or our hypothesis. And I'm going to skip this one, but I have some interesting ideas about how these models can be updated and augmented to dissect the interplay between visual attention and decision making. And third, I'll be leveraging this technique to enable precise external control of neural population dynamics in behaving animals. So one of the holy grails of neuroscience is to be able to manipulate the activity of a neural circuit, arbitrarily specifying the neural population state. So if a neural population state is here, I want to press a button and make the state move to here, say. And if you could do this, you could precisely test most any theory about circuit function. So most of the talk has been about what we can read out from the brain, but here the idea is to precisely write into the brain. There are many technologies for perturbing activity of neural populations, including electrical stimulation and optical stimulation with optogenetics. And the idea here is that because we know how to build rich models of neural population dynamics, we can bridge with techniques in model predictive control to identify the electrical or optical simulation procedures that optimally guide the neural population to the desired state, all while taking account the neural population dynamics. And some potential applications of this could be to encourage a neural population to explore new patterns of activity during learning, which could be helpful for stroke rehabilitation. Or it could provide artificial tactile feedback from a robotic limb. Or dare I say it, it could teach you Kung Fu. And so with this, I'd like to thank my wonderful advisors and collaborators, and I'd like to thank various funding agencies who've supported my work over the years, in particular, the NIH for funding my K99 or 00 Faculty Transition Award, and I'd be happy to take more questions.
All right, let's thank Matt for the great talk. And I think, yeah, we have time for maybe a couple of questions. Looks like especially Edgar Walker has been quite, has quite a lot of questions. I don't know, Edgar, do you want to pick one of your questions? And let's go with that.
First of all, very exciting talk. I really enjoyed it. I threw a lot of questions, but perhaps I'll stick to the last one I threw. So for example, describing the dynamics and like understanding, like capturing with the recurrent neural networks, I'm curious, have you considered applying for like theories of probabilistic computations, for example, like perceiving the dynamics that occurs as like a manifestation of, say, sampling that will be predicted from sampling theory? Like, is that kind of work that relies along your interest? I'm asking because that's personally very much in alignment with my interest. I'll stop there with the first question.
Yeah, that's a great question. I think that's another really interesting, maybe higher level computation that, yeah, many have theorized that the brain does and have shown some evidence for. So I think, yeah, it would be fantastic if we could use these techniques to start looking into that type of thing. How does the brain perform statistical inference?
Mm-hmm. And I'll maybe just throw one quick question and then I'll go off. Like in terms of the recurrent neural network, so like the result you've shown so far has been on synthetic data, I see, right? Like in the synthetic data alone, like if you're producing the spike count simulation and then retraining with the recurrent neural networks, like what's the accuracy with the prediction? Like, are you able to like capture the response like extremely well, or do you still actually find issues with it in terms of like achieving good predictive performance?
Yeah, we can sort of arbitrarily control the answer to that question by having more or less noise in the network, or having more or fewer neurons that we simulate in this process. So I think absolute numbers are not super helpful here. But I will say, you know, when I go between modeling the synthetic data at any noise scaling to real data from the brain, it's really humbling, because there's just a lot that we don't understand about the brain, and the brain doesn't just take one input about the thing that you're looking for, but rather, you know, other cognitive factors are driving the activity in the brain. And so it's important to, I think, take advantage of or factor those in and build components into the models that can factor those in.
Absolutely. You didn't have any data that you presented, or maybe I just missed up on applying this model directly to the neuronal response yet, right?
That's correct. Yeah, I only kind of briefly mentioned some preliminary results that are not quite ready for prime time yet, but soon.
Understood. Awesome. Thanks so much.
All right, thanks, Edgar. I have a quick question, Matt. I guess I'll invoke that right as the moderator here. One of the things that you know, people want to know, I would think here would be, you know, you're suggesting getting insights from the brain in terms of you know, computation in recurrent neural circuits, and so how do you think those principles would apply in a broader context in terms of AI, for example, designing AI you know, frameworks? Do you think that the gain is in terms of explainability or interpretability, in terms of the fact that you have these motifs now and you can start to understand how these complex recurrent dynamics are proceeding? Or is there some other gain one would get as opposed to just training like a LSTM or some standard you know, a transformer network that would solve the task, right? So what do you think are the big gains you hope to get that would apply also to AI, if any?
Yeah, I think this is a long game. Most of the work that I've described today is really machine learning that's been enabling the neuroscience. For example, this fixed point analysis was first, pioneered on machine learning networks that were, didn't really have so much to do with the brain or brain data. And now we're trying to bring that into neuroscience even more by applying it to neural data. And this is kind of a common theme along a lot of things. Machine learning research moves so fast, and neuroscience research and biology is really slow and hard. Not that machine learning isn't hard as well, but there are many things that we don't control in a biological system, and we're kind of flying blind in many cases. So I think it's a long game, because there are also just components of biology that we don't even know how to really think about in terms of machine learning, things like consciousness, for example. So I don't know if maybe in the longer game, once we start kind of breaking down the building blocks of the brain, maybe some of these really, really challenging concepts to tackle in machine learning will kind of come out as emergent properties.
All right, great. I think Michael Nolan has a question. Michael.
Yeah, thank you. Great talk. I have a question about the task where you were fitting the LFAS model to the task RNN you had already trained. So I know like LFAS takes spikes in and nominally produces spikes out like through estimated spike rate. So I was wondering how you were producing like just these spike rasters from the task RNN. Like was that like some spiking RNN model, or were those just simulations derived from like the spike rates computed as like the actual RNN unit activity?
Yeah, so it was the latter. These were not spiking RNNs, but we wanted to fit LFAS to data that looked like the type of data that we record from the brain. We wanted to fit it to spikes. So we simulated spikes from the hidden units of the task RNN, and we did that by using the rate scaling the rates in different ways to be the rates of inhomogeneous Poisson processes from which we could then sample counts from.
Okay. So
representing spike counts.
Connects, quick follow-up, sorry, connects to Edgar's previous question. So the like the factors or the generator outputs that you're seeing from LFAS trained to recreate that, does that activity look like the task RNN activity?
Yeah, it looks, it does. Yeah, that we're optimizing for it to do that. So I think that's kind of, yeah, that's the goal of LFAS. And what's a little bit less of a given is are the dynamics needed to generate that the same dynamics that generated the task RNN, that are the task RNN.
Cool, thank you.
Does that make sense?
Last chance for questions. Okay, Eric, go ahead. Eric Shia Brown.
I was wondering if you could kindly seek Matt to connect the two parts of your talk by perhaps positing what it was that was changing in the within manifold learning that took place over hours time scales in the reassociation paradigm. Could that have been the inputs, for example? And if so, is that something that you can learn with LFAS or other sort of dynamical systems models of underlying circuits?
Yeah, that's a great question. I think I wrote a bit about this in my research statement, which is that, yeah, we'd love to go in and fit LFAS models to the population data that we recorded before learning, and then selectively optimize different parts of the network to get it to produce the data that we saw after learning. And so I think this is the way to address your question in our rate networks, which is, can you get that? Can you get LFAS to reproduce the after learning data just by reoptimizing the inputs? And I think the answer is yes for reassociation. And I think the answer is no for realignment or anything that happens outside of the manifold. I think for that, you need to have a reoptimization of the connectivity within the network.
That's fascinating. Thank you very much.
All right. Again, thanks again, Matt, for a very fascinating talk. And I think, yeah, we'll wrap it up now. And again, thanks to the audience. Great questions, by the way. Thank you again.
Wonderful. Thank you all.